My name is Nicole Mackey, and last June, my mother, my 9-year-old son Devin, and I were honored by being asked to take part in the filming of a Barth Syndrome Foundation PSA while attending the Barth Syndrome Clinic at the Kennedy Krieger Institute in Baltimore, Maryland. Today, it is my distinct privilege to introduce the final product, a video produced by the Bird's Nest Foundation, to all of you. Weeks old. We had no idea he was sick.
We didn't know that Kevin had Barth syndrome until he was 7 years old.
It's claimed both of my sons' lives, but it doesn't mean that it's defeated me.
At times you wonder why me and feel a little isolated, but at the same point, you know, he's here and it's a blessing, so you just go on as a normal life you can.
Barth Syndrome Foundation is the only foundation in the world dedicated to helping families and individuals who've been diagnosed with Barth syndrome.
In 1996, I took my son, Michael, to Amsterdam to see Dr. Peter Barth, who originally described the disorder. And he said there are others and you should find them, because if you find one another, you can help them. I started a website, and I didn't know anything about starting a website, but one of the things that I've learned is the impossible is possible.
Barth syndrome is a genetic disorder that occurs in boys.
Barth syndrome was described for many years as children who have a certain type of heart disease, low white blood cell count, and some growth delay. And it was only in the mid-1990s that a particular gene was found to be the cause of Barth syndrome.
It waxes and wanes, it gets better and it gets worse. And I think that's something that's unusual, something that's kind of unexpected.
When I was about 2, I had a cold, and my mother picked me up and just holding me in the night and realized that my heart was beating very, very fast.
We had a child with weak muscles, and now we had a child with a heart that might need to be transplanted.
And that was when we ultimately discovered that I had Barth syndrome.
The first thing that we went through was the 12-week course of ICU, him having a heart transplant, and 6 months later we finally got the diagnosis.
Even though he's just laying in a bed all hooked up, he's still there with us, so but you just hold on to every moment you have with him.
I don't think you can overemphasize this sense of isolation that parents have at the outset.
There was no one else in the entire world that we knew that had it. And when he finally got a diagnosis, Dr. Kelly gave me one phone number.
At one point I was being followed by 10 different doctors. There were some months I'd be there every week.
You walk into an emergency room and try and explain to a doctor who's never seen you before that even though you don't look very sick, you're extremely ill. Exhaustion is a daily prospect. I'm in bed a lot of the time. Pain is a huge issue. Constant doctor's appointments are a problem.
We went for an overnight one night, and the other little boy just got up and ran to the car. Well, Henry had, I think, 9 different medicines and supplements to take. And so you don't realize how much time you spend doing all of the medical side of things during the day until you see another healthy child with them.
Why is this happening? Why isn't he eating? Why isn't he growing? Why isn't this happening? Why isn't he doing what my other son did? All those questions were truly never answered until he was way beyond that point.
Probably around 5th, middle school age is when the differences between people with Barth and regular kids start to show. They're growing at a normal rate and you're not.
All the boys end up reaching normal height, but during their early school years, they're the smallest child in the class, often smaller than some children 2 grades back, and it's difficult socially.
Barth definitely has social ramifications. Particularly as a young man, you're judged, particularly by your peers, as a physical specimen. You know, how fast can you run, how high can you jump, how well can you play football.
Every new thing that he can do that
that he's probably been delayed on. It's a mark for a celebration.
What's unique about Barth Syndrome Foundation is really the sense of community and the remarkable things that they've been able to accomplish in a relatively short amount of time with few resources.
It just makes me not feel alone. I have people to go to.
You can look into another mother's eyes and they know.
It was a resource, it was a mechanism for us to have more hope about his future.
We collectively believe that one of the greater partners in care happens to be the parents. And when a parent begins to trust their instincts, then they are empowered as a partner to make a difference in their child's life.
We have stem cell research going on up at Harvard. We've created a Barth mouse, which can serve as a mammalian model, gene therapy. And finally, we have a very well-attended international scientific, medical, and family conference that happens every 2 years that we fund entirely.
It gives us a chance to connect with not only other families that have children or young men affected by Barth syndrome, but with doctors who have experience with the condition.
The foundation recruited just about every important person, so it really is a model for how a family disease organization should work.
We're accredited by the Better Business Bureau. We're a member of the National Health Council.
We all feel a tremendous responsibility to find the other Barth boys who are out there. If we don't do it, these boys die. If we succeed, they don't just survive, but they flourish.
The thing I say about rare disorders is they're not rare to the person or the family that's affected by them. For them, it's their whole world.
It gives me a chance to tell people about my children and tell people about this rare condition that he has.
We've made so much progress that has made the little kid's life a little bit easier, but there's more to be done.
There's a lot more out there that are undiagnosed, we're sure of it. We want to gain more knowledge and to help these boys live longer lives.
A doctor said to me, the difference between a Rottweiler and a Barth parent is that eventually the Rottweiler lets go.
We're not ever going to give up, and we can ultimately rid the world of Barth syndrome forever.
Barth Syndrome Foundation is hope for a treatment, for a cure, for peace of mind.
We would love for you to join us.
You can make a difference.
Join us.
Please join us.
Thank you.
Thank you.
Thank you.
So I still tear up. My first son passed away when he was 2 weeks. Okay, that doesn't help. I still tear up each and every time I see that video, partly because the stories of personal loss are ones that are far too familiar to far too many of us in this room. But even more so, my tears are those of hope. Thanks to the diligent efforts of the Barth Syndrome Foundation, our boys are now living longer, growing stronger, and as my personal favorite part of the video says, they now flourish. But while BSF has already done so much, there is still so much more to do. To quote our mission statement, the Barth Syndrome Foundation is dedicated to saving lives through education, advances in treatment, and finding a cure for Barth syndrome. Each and every one of us has an opportunity to propel the mission at this very moment. Donation envelopes can be found on every table for those of you who wish to contribute to BSF, to continue the great work mentioned in the video and going on right here at this conference. The foundation has set a conference fundraising goal of $75,000. Please help us surpass that amount, and in doing so, secure a promising future for our beautiful boys. Speaking of our beautiful boys, I've been asked to introduce someone who I remember as a boy, specifically as a boy playing the lobby piano for tips 4 years ago at the conference in Orlando. Apparently, 4 years is the amount of time it took to turn that boy into a young man. Friends, please welcome to the stage R.J. Kugelman.
So when I first heard Barry Byrne was doing the keynote, I was all like, dibs, I am introducing Barry. I don't care who anyone else is. Barry is mine. So slide 1, Kendall. So we don't actually know where Barry is right now. Last time I saw him, he was parasailing, and he said he'd be back in like an hour, and I don't know where he is, so whatever. Next slide. So last conference, if you guys remember who you were here, Barry Byrne was not here. He was climbing Mount Kilimanjaro. And this picture is actually from Teton, and he climbs a lot of mountains, and I don't really know why you would want to climb a mountain, but he does, so that's his choice. But, next slide. I met Barry when I was 3 weeks old. Barry was the cardiologist on standby when I was airlifted into Shands. And that picture is actually from the 2004 conference at Disney. I don't remember what hotel. But I was actually not the first Barth boy Barry ever met. In, I don't know the year, but when Kevin Boffa was a baby, they called him Baby Boffa. That's what Barry called him. Barry actually treated Kevin years ago before anyone even knew about Barth Syndrome. Before Barry knew about Barth Syndrome. Before Kevin knew about Barth Syndrome. So it's kind of crazy how here Barry comes just randomly. Next slide. So the thing that makes Barry special is he's more than a physician. He really just cares about the patient. He always puts the patient first. And here we have, this was my original team when I first started going to Shands, with Barry, Dr. Bryant, Dr. Spencer, and Michael Bowen, of course. But Barry has always been the one out there dancing with all the kids, just always so crazy and has such a great personality, and that's really what makes Barry a genuine person. Next slide. All right, so there's actually a story behind this picture. Barry kept wanting to get a skin biopsy for me to get some stem cells from to grow, I think a heart muscle is what he's growing. And so I made him a deal. I told him that if he takes me to a Gator game, he can have a piece of my skin. But what I didn't know is how difficult getting that skin would be. Each time we tried to get it, there was something wrong with the consent forms, or if they didn't have one form, or something was messed up. But finally, it happened, and they took my skin. And then we went, he said we're gonna drive up there, and he's gonna have like a barbecue at his house, and he's gonna be parking cars and stuff, because that's what he does for fundraising. So we go up there, and we're driving on the way there, and he calls me, and he says, change of plans. I ran into the president of the PAL gene therapy, and me and you have a seat up in the president's box now, so your parents can have our tickets, and we're gonna be up in the president's box, and they can have my seats. And I'm like, okay, whatever. He's like, I'm running to the grocery store right now on my bike. I'm gonna buy some food for a barbecue. Like, okay. So we're driving up there, and we get there, and Barry's out there parking cars, soliciting, and we go in his house, and then his wife walks in and just looks at us. Apparently Barry did not tell her that we were coming. So there's just some people in our house that she doesn't know. And so we're like, so Barry, when's this barbecue? He's like, oh, I didn't have time to run to the grocery store and get food. So we go to the football game, hungry, and while we're there, we're in the president's box, so they have like turkey and cranberry sauce and like Thanksgiving dinner practically. And we decide we're going to bring my parents food because they're just having a hot dog. So we get food, and we're bringing it down, and while we're in the elevator, Barry starts to eat the food. Like, Barry, you can't eat this food. And we get there, and we can't find them. So we just go back upstairs and eat the food. So that was, I think that's like the only picture we have together. I don't know. But the president's box, you can't see it in that photo. It's right behind us, actually. All right, next slide. So this is just some of the stuff of what Barry actually does. And I think the interesting thing about Barry is when people really think about Pompe disease, I think Barry is the first physician that they think about. I mean, he was the medical advisor on the movie, I can't remember the movie's name. Extraordinary Measures, that was it. He was the medical advisor on there. He had like his own director's chair and stuff, advising that whole movie. He actually had a cameo appearance in there. We saw him. He's the little one in the blue shirt walking in the back. But he's someone who many other physicians consult with throughout the time. And it's just amazing how Barry Byrne is a name and people know it, and it's crazy, I think, that we even have him. Next slide. So this is Barry, dressed as a Spice Girl. This picture I did not have. Thank you, Dr. Brian and Connie Nixon, for this photo. That was lovely. Thank you. And here's a little story about Barry. 2 months ago, I was at my sister's graduation, which was outside at like high noon, and it was really hot. And they split the alphabet to where K was like at the end. Well, I made it to about D, and then I nearly passed out and was thrown on a stretcher in an ambulance to the hospital. Luckily, the hospital was right there, right on campus, like 5 minutes. So the first person we call is Barry Byrne. And Barry's like, oh, I know a cardiologist over there. I'll call him. So he calls him, and we get there, and some cardiologist comes in. He's like, are you RJ? I'm like, yeah. He's like, Barry Byrne called me, and he says, starts talking. I don't know what he's saying. I don't understand cardiology speak. But he starts telling me all this stuff, and Barry's on top of it, and that's what really makes Barry special. He's there when you need him. He's great. And next slide. This was, I think, my first time at Camp Boggy Creek when Barry was there, and there's been a few. I've been to Camp Boggy Creek previously, and Barry has, this year he was in Spain. Last year, I don't know where he was, but he was not there. But this was at the dance, and I was amazed how Barry knew all the dance moves that all these kids knew. I didn't even know any of these songs, but he knew everything. And that's Barry. He's a real special guy, and I would like to bring him up now.
This is lovely. Thank you, RJ. Well, it's a real honor to be invited to give the keynote address for this BSF conference, which it really has been a journey, as RJ said. And thank you, Shelley, for all that you've done introducing me to this great family of kids and parents. And the title of my talk today was, in a way, to give you some insight on a few examples, including those that have been done recently in the lab with an intention of developing a treatment for Barth syndrome under the idea that there are really some new opportunities now for drug development for orphan diseases, and kind of encapsulated in this Time magazine cover about knowing your future. Everyone wants to predict the future. Sometimes the future can be insightful. Sometimes it can be a challenge, and you see that it says new genetic tests can describe a given gene, but they don't necessarily point to a cure for a disease. But I hope I'll be able to tell you that this knowledge is really something that we gain from understanding the DNA that programs all of our cells to function normally. That knowledge has actually advanced so much since the onset of the Human Genome Project, and now in this genomic era, this information, this simple code, is really the information we need to design specific, impactful therapies for genetic diseases. Iris Gonzalez crafted this great depiction of what the sweater gene looks like. You can see on the left a well-designed sweater may look something like this on a child. Then there can be small changes that might be a single point mutation that may not have a major impact, but if you see on the right, sometimes there are major problems where the hat and glove wound up sewn into the sleeve, and that sweater might look like this. It's a bit of a problem. So what's driving this is the fact that the Human Genome Project, at $3 billion in 1993, the information to get all the information in one single genome is now in 2013 just $900 to get that same bit of information. So we can use that to develop personalized medicines, and the personalized approach as it relates to genomic medicine is really to develop a more precise diagnosis and target the optimal population with the treatment, and then eliminate all the unnecessary or empiric treatments that sometimes involve drug reactions or poor compliance with therapy. And in the end of the day, this will improve patient outcomes and ultimately lower healthcare costs, which we know is critical at this time. And one of the drivers of that is the Orphan Drug Act in 1983, which provides a benefit to sponsors who develop drugs that are effective in populations less than 200,000 people. And that gives them protection against other competitors developing a similar drug. And this actually came to fruition just this last year. The orphan drug designation for Glybera, a drug developed in Europe, is a gene therapy, the first gene therapy approved in the Western world. This product was, in July of 2012, finally awarded to Amsterdam Molecular Therapeutics. And this is their letter from the European Medicines Agency. This gene therapy approval actually sparked a wave of investment in the past year with this strategy for treating orphan diseases because of the findings of the impact that this had in this one condition. This is based on work we did originally in 1996, and you can see the length of time it took to reach approval. We used a different example back then, but this was focused on delivery of genes to skeletal muscle. And in this example, the outcome was looking at hematocrit. But I'm going to show you some examples of other neurological and musculoskeletal conditions that are relevant to Barth syndrome. And the idea behind this is that in the conventional drug development world, one might take tens of thousands of compounds and test them one at a time to try to identify a few that could be used in the clinic, and ultimately one drug is approved. But you can see the timeline. This really just takes too long. And sometimes that path is very crooked. But if we had one gene that is the cause of the condition, we hope that that's a more straightforward pathway, and in fact we can eliminate a lot of the drug discovery steps. So there's 3 years taken away, and that the clinical trials required to show an impact of these specific therapies are also much shorter. In fact, in the past few years, each of these studies has been as short as 6 months because the impact is so high. So I'm going to show you a few examples of studies we've done at our institution in collaboration with others around the world. And one of the most impactful has been for Leber's congenital amaurosis, a form of inherited blindness. There are about 400 single gene defects leading to premature vision loss. And in this condition, we used a small delivery package for the gene for LCA in adeno-associated virus. So this is a schematic of AAV. It has very specific receptors for different cell types, and this version, AAV2, was used to deliver the gene into the eye. So here's how it works. Through a surgical intervention, the surgeon places a needle into the back of the eye, and underneath the retina delivers the gene within the AAV. It enters the cells, makes its way to the nucleus, drops off the gene, which is made into the therapeutic protein, but it's most importantly specifically in the cell that needs to benefit from that gene defect. And one of the first patients to receive this therapy was Dale Turner.
So I took off my sunglasses and I peered into the sky, and the sky was something I had never seen before. In that, I mean, the color of the blue was just overwhelming. I mean, it was something that struck me, and it was a very emotional moment. It was just the proof was right in front of my eyes, literally.
So Dale was a brave fellow. When he came to Gainesville, he was supposed to travel with his dad, and I went to pick him up at the airport, and there's this young man with another gentleman who seemed like he was his dad, and they were chatting, and Dale had his 2 bags. And keep in mind that his vision is so impaired that even the number of the gate is something he can't see without some help. So I introduced myself, and I said, good to meet your dad. And he said, no, that's not my dad. My dad had to go on a business trip. I just met this guy in Atlanta. So he managed to make his way from Toronto to Gainesville on his own, and he's a very brave young man. He's now graduated from law school and practicing law in Toronto. Another example is a study we did with investigators in Taiwan. The group in Taiwan at the National Taiwan University studies a type of movement disorder called congenital AADC deficiency. This is a huge challenge for these infants because they make no neurotransmitters. And so they really have no movement, and some paradoxical movements of the eyes in particular. And here's a video of one of the children preoperatively. You can see that they're really not able to interact with their environment, barely able to smile. And so Paul who was the pioneer of this idea, we worked with to develop vectors that were injected into the part of the brain where these neurotransmitters are needed most. And after the surgery, you can see the stereotactic delivery of the missing gene into the center of the brain, into the putamen. And in fact, after the surgery, you can see all the children that were treated were now able to sit on their own and even smile. So this was a really huge leap for this patient population, and now he's continuing this study in the US. I'll give some examples of the, as RJ mentioned, we've done a bit of work over the years in a form of muscular dystrophy known as Pompe. And the Pompe story, as he mentioned, was told in this movie, so if you want to know the facts, you can watch the film or maybe read the book. It's actually probably more detail. But in the movie, there's I think a very touching moment when the family learns that their child can't be helped anymore by the conventional treatments.
As you know, Pompe patients suffer muscle deterioration in every part of the body, so this sort of respiratory complication, well, that's just an inevitable fact of the disease. Mr. Crowley, Mrs. Crowley, Megan is not responding well. Okay, now we've done everything we can, but she's just, there's nothing more we can do. I'm sorry. As you know, she was already well past the life expectancy for, Pompe kids. After all, it's not just her lungs, okay? Her heart, her liver, these organs have been compromised and would have become fatally enlarged. Now, I wish that we had a drug to treat Pompe, but we simply don't. I'm so very sorry.
So those words, "I'm so very sorry," are really something you never want to have to tell a parent. But they really captured the frustration of that time, and there really was no treatment. But really with the pioneering work of a number of laboratories and the sponsorship of Genzyme, a drug was developed, and the goal was to eliminate a glycogen buildup within the muscle cells, so another kind of metabolic disease. You can see the impact on the heart. Really, this was probably the single cause of why this was such an early severe disorder. And you can see an example of a chest x-ray in a newborn affected with this condition, and then following the treatment, a huge impact on myocardial size. Now, not every patient responds so well, but it did lead to a really huge impact in mortality. You can see the treatment group on the top line in blue, and the natural history control, in which only a few % of the children survived out to 18 months. So this led to the approval of a drug, and this was actually the first cause-specific treatment for any neuromuscular disease. There's still a lot more to do, and that has led us to investigate another approach in which every cell that needs that protein can make it themselves through transferring the gene for alpha-glucosidase into the cell. And we use that same virus that I showed you in the experiment with Dale. AAV can be delivered to muscle and nerve cells, and it, through the work of Catherine Ma, shown here, and then Christy Pasek, who's here at the conference, also helped develop versions of this AAV which are highly tropic for the heart. And I'll show you a little bit about that and the study we've done in the Pompeii group. What we learned in those patients that survived over a year is that they had progressive loss of independent breathing. So we really wanted to target the diaphragm muscle. So in a study that's just been completed, we looked at diaphragm function both in a control, you can see here, and in a Pompeii patient on the right, and you see the defect in diaphragm function, and this is what leads to ventilator use in the patients. Darren Falk, who began some of that work in the diaphragm, has now determined that one of the defects in this disease, which we didn't know when the patients hadn't survived past 18 months, is in the neuromuscular junction. And so the nerve cells also have glycogen, and the only way to reach them is with a gene therapy approach. And so Darren and others in the lab showed that the vector itself could restore nerve function after delivery to the heart and lungs. And so in the clinical trial that followed, we evaluated both the effect of this gene therapy on the neuromuscular junction, but also on respiratory function. And this is work done by Manuela Corti to show that actually in affected and healthy patients, there's a difference in the characteristics of the neuromuscular junction in a function called jitter by single fiber EMG. Barb Smith is also a physical therapist who studies respiratory function, helped characterize all the patients in the clinical trial. And you can see the unaffected patients have very regular and robust breathing flow patterns. And in the Pompeii patients, there's very diminished and irregular flow patterns. And what we did was to try to strength train the patients, and you could see that there was no effect of strength training itself, because that requires voluntary control of the diaphragm. But after the gene therapy itself, all the patients in the study gained some function by increase in spontaneous ventilatory endurance. So we now want to move this trial to a more systemic approach, because more muscles than the diaphragm are affected, and you'll see at the end why this is now probably relevant to Barth syndrome. So to move towards systemic therapy, work Darren did was to show that we could actually achieve a level of expression that was at or above that of a carrier, which in Pompeii there are unaffected. So whereas the enzyme replacement therapy didn't have the same effect on the activity in the heart. So that concept then to give a single drug that's effective against the disease gene is where we are in many of these conditions, and we think that this very long drug development pipeline can be shortened. Here's an example of what it takes in the systemic dosing study that's planned for Pompeii. all the way back in October, we began the IRB submission, met with the FDA in December of 2013 to plan the filing of the IND, and in fact the final IND submission will be in November. So this, in every instance, takes a lot of planning and preparation to go through this regulatory path. And this is the timeline of one of the AV9 projects, where you see that the preclinical material developed in December, and not until January 2015 will the clinical trial begin. But all these steps need to be followed to ensure one of the really important tenets that the FDA proposes, that all drug studies be safe, and that and to reach approval they have to show that they're effective. So here's where I think we are with Barth syndrome, just to end. Megan Sostek has committed her graduate career to understanding this problem, and she's also here. She really was the prime mover behind characterizing the TAZ knockdown mouse, along with the work done by Kuchka and Arnie Strauss. Megan showed that the short hairpin RNA induction causes significant knockdown in TAZ, and you can see how little tafazin is actually produced in the key components like the heart muscle and in the skeletal muscle. That has an impact on cardiolipin remodeling, and the aberrant cardiolipin profiles in the knockdown are shown in the bottom panel. So that gives us one biomarker. This mouse is not as severe as in the patient phenotype, so we continue to look for more information that could uncover abnormalities. The mitochondria are clearly abnormal in the model, but that's also a difficult clinical endpoint. She characterized the contractile force, and so shown here in a muscle bath is some of the muscle from the Barth mouse being stimulated to contract. You can see, just like we found in the patients, there is a defect in force. And then loss of tafazin does impact heart function in the mouse, although it's subtle. Colin Foon has done some additional work in this and shows even early on there can be even more profound defects measured in the right way. So we think there is some value to the model, but now our challenge is to develop, again, the specific therapy is where we're at. One of the things Megan tried to do was exacerbate the phenotype by exercise, and Cade talked a little bit about exercise today. This is actually more of a survival test than exercise training, but these are a bunch of Barth boys swimming to save their lives. And one of the unusual results of that was really unexpected. I think you'll ever see any of the boys in the pool doing that. So the endurance training did have an impact, though, in the mice. It really increased left ventricular mass. It had an effect also in the energy utilization, and we think in mitochondrial number. So her next challenge was to see whether we could replace tafazin initially in vitro models, and now ultimately in the mouse model. So we designed a few vectors like the ones I showed you earlier, ones that are controls. So here's one that expresses green fluorescent protein, and then two different promoters, either the endogenous human tafazin promoter driving tafazin, human tafazin itself, or one that's restricted in expression to heart and skeletal muscle. And when she infects those constructs into TAZ-deficient cells, you can see that the vector produces a protein which localizes to the mitochondria like it should. And then importantly, that these constructs, these specific therapies for Barth syndrome, restore cardiolipin remodeling. So on the very left, you see the very low monolysyl cardiolipin to cardiolipin ratio in control cells, and the abnormal ratio in a Barth fibroblast that has been converted to a muscle cell, but treated with the control non-TAZ construct for GFP, and then on the very right, the one treated with what would be a clinical candidate for treating Barth syndrome. So the options now to take such a therapy forward for a rare disease drug approval have changed in the recent years. And this is what I hope to leave you with as a message of hope as to about how sometimes you feel like you're just too small to reach the attention of that group that may be able to help develop a treatment. But there are opportunities now through legislation that is the Prescription User Drug Fee Act provides money back to the FDA from industry to accelerate the approval of drugs for certain indications. So this recent iteration of PDUFA actually has specific language about rare diseases and developing patient-reported outcomes, which you heard about also earlier today. And so the key points from this are two categories called accelerated approval and breakthrough therapies. The breakthrough therapies allow an expedited approval based on a biomarker for a rare disease, and in something that takes many years to develop, that kind of discovery can really expedite the approval process. And this is specifically designed for life-threatening conditions. So what I hope you'll accept is that we now have a mechanism, given a well-defined natural history. This is the value of the registry that many of you participate on, and the potential to have a very large drug effect like a specific gene. It's possible to design a single open-label study that can be the basis for approval, and I think that's actually an option in Barth syndrome. So just to summarize, there are many rare diseases, really over 7,000, affecting 25 million people in the US. A lot of orphan drug designations up to 2012, but only 400 of those have led to designated products. So there's really a lot of room for improving the success of developing what are really need to be sustainable therapies by approvals. And so of those many diseases, only 250 with treatments. But I hope that you take home that the strategy that we've worked on will be very effective by providing a very specific treatment for the root cause of the problem or the genetic abnormality. And I just want to say thank you both to all of the funding agencies over the years, and both our clinical team and the team in the lab. It does take a group. It's not something any of us do individually. And we particularly want to thank the patients. So here's the spectrum of patients we see with Pompe. I also want to acknowledge that we've made some progress. This is the first patient that we have with that condition that is now able to walk after many years of trying different approaches. And I particularly want to say thank you to Shelley for all of her guidance, and as this picture has been in my office since I met Michael. And to RJ. So thanks for the introduction, and thank you.